I hope you all had some coffee and some sugar so that I will not put you to sleep. Here's a brief outline of what I'm going to try and achieve today. As Susan mentioned, my goal was not to present a larger overview of the field, but rather to really give you an update of what's new since the last time you might have come to one of our symposia, what happened in the past year. So I'll very briefly introduce just a couple of the measures that I'll be talking about, and then I will give you a brief research update on a few studies that I found the most interesting with regard to the reward system, this important network in the brain that we and others have found to be impacted in autism. I will also tell you about a few studies on oxytocin and its influence on the social brain. I will then switch gears somewhat and talk and give you an update about new findings in the field of brain connectivity. In particular, there was a big fuss about emotion confounds, which may or may not have made it to the out there to the larger community. I'll also tell you about some new data that pertain the issue of whether the autistic brain is better characterized in terms of as being underconnected, overconnected, or perhaps a bit of both. And then I will also conclude by highlighting a few avenues for future research, talking about at least one of the studies from our lab that came out in the past year, something new in the realm of data analytic approaches, what they can offer, and also some of what I think we will be seeing in the future. Okay, so I guess everybody has seen these before. This is what you would get from a traditional MRI. You get a very nice picture of what the structure of the brain looks like, but for the most part, what we have been interested in is to actually look at brain functions. So rather than focusing on the structures, we are focusing on this cute large blobs, color blobs that indicate areas of the brain that are active or engaged when a person is performing a variety of tasks or when they're presented with some particular stimuli. So when you see a picture like that, those blobs, that's exactly what they typically refer, patterns of brain activity. However, in this particular case, these blobs here were not observed when a person was presented with some auditory stimulation. They were actually generated by picking a random voxel, focusing on the activity on the signal of one of the voxel in this region, and extracting his time series over time, what were the changes and fluctuations in signals, and then asking wherever else in the brain there may be activity that mirror the same pattern of activity. And what you see is that this other, the contralateral auditory cortex in this example, has activity that is very highly synchronized with the activity of the auditory cortex in the left hemisphere. So again, this was not collected during a functional MRI task when they were presented with stimuli. This is during what we call a resting state scan. So we use the same techniques. The person is still in the MRI, and we're collecting our MRI data, but we're not asking them to perform any task. Nevertheless, there are spontaneous fluctuations, as you can see there, in activity, and that's what we capitalize in order to obtain maps of the brain of networks that we actually that have been termed intrinsic connectivity networks that yield a measure of a functional connectivity, how the brain is wired, if you will. So what you see here is it's a series of pictures that shows several of these intrinsic connectivity networks that are identified using this method while the subject are at rest. And these same networks can be observed both when a subject was at rest, but also if they were asleep and also if they were under anesthesia. So what these maps basically tell us is that they tell us what regions tend to work together in order to subserve some cognitive function or task. And so they basically reflect the history of a co-activation between regions. Now, this functional connectivity measures that I'll be telling you more about stands in contrast to structural connectivity measures, which I will also mention briefly in the rest of the talk. And here, you don't see that, you still see fairly broadly colored images. That's what neuroimagers like. But we're not talking about the same blob, because here we're actually measuring tracts. We're actually measuring those structural physical connection, those white matter tracts in the brain. And if in case you cannot quite see it, this is your spine, and this would be the cerebellum. And this is, I think, a very artistic picture of a single person DTI, diffusion tensor imaging study. And I put this also as a plea for anybody here who's interested in participating in our studies. When you actually do get pictures of your brain, not just this boring structural pictures, but you can get one of these too, you or your children. So that, I hope it's an incentive. Okay, never mind, moving right along. Here, I told you I was going to start with telling you a little bit more about some recent reward studies. We have done some early work. I think we were the first to publish evidence of hyper-responsivity, reduced responses in the reward circuitry. But since then, there have been a flurry of other studies. Our study was published in 2010. Here's an interesting one where they actually looked at how the reward circuitry might have been modulated by whether the pictures that the subject were presented were depicting some activity that the particular participant were really into. And lo and behold, I mean, I don't have time to go into the details of every study. I'll give you kind of like a survey. But what was interesting here is that they actually found not so much differences in the core of the reward circuitry, but in some regions like the anterior insulates and the cingula, which are part of the Salience network, which is also a network that I'll mention in some of the future studies. It's one of those intrinsic connectivity network that it's supposed to direct the attention to what is most relevant. Sometimes it may be stuff that happens in the environment, or sometimes it may be also something internal to your body states, like whether you're hungry or sleepy and so on. There was another study that used fairly similar stimuli to the one we used, and they found again hypo-responsivity, hypoactivity in the reward circuitry in individuals with autism. But to both social stimuli and monetary rewards, we found somewhat less of a response for social stimuli in particular, but as they admitted themselves, our stimuli were more rewarding because we used both verbal praise as well as a smiling face, whereas they only use a face. So. But my favorite in this series of studies on the reward circuitry is actually a study that it's a connectivity study, a functional connectivity studies, where they found some very interesting relationship, or I would say lack of relationship. We all know that lack of attention to the human face, as well as a lack of preference to listening to the human voice, is one of the markers of autism, and one that is seen fairly early on. We also know that very young babies fail to orient, for instance, to their own names, children that are later on diagnosed with autism. We know that older children with autism do not prefer to listen to motherese or that high-pitched mother tone speech. They rather prefer to listen to a computerized, synthesized speech. And we also know that in older individuals with autism, two regions in the brain, in the temporal cortices, that are really showing very specific responses to the human voice, fail to show this strong specific response to voices. But nobody has yet related this lack of sensitivity to the human voice, both either behaviorally or in the brain, to the reward system. And that's what this study did. They basically zoom into those two regions that I mentioned in the posterior superior temporal sulcus that have been shown in several studies to have this very strong preferential response to the human voice. And then they seek to identify which were the regions where this with what other region these two hubs were connected. And they did this in a sample of individuals with autism and in neurotypical controls. And lo and behold, what the pictures that I'm showing you here is showing actually all of the regions where the typically developing individuals show greater functional connectivity than the individual with ASD. And I don't expect you to, extract much information from this, but let me highlight that the region that you see here showing a very strong relationship or being very highly connected to this voice selective regions are really at the core of the reward circuitry. So you have the nucleus accumbens, you also have the ventral tegmental area that shows activity, the putamen, and there are also a few other regions, like I mentioned the insula, for instance, as being important in the Salience network. You also may see some activity in the medial prefrontal cortex, which is one of those theory of mind regions that study after study in the neuroimaging literature have found to be important and typically showing hypoactivation, less activity in individuals with autism. And a similar pattern of finding was also found when you were looking at the connectivity with the right superior temporal sulcus regions. In case you prefer to look at data in a different forms, as you can see here and just in bar formats, you can see that this effect was incredibly robust across all of the regions. So typically developing individual having much stronger connectivity between this voice regions and the core of their reward circuitry, which suggests that indeed listening to the human voice may not be as salient or as rewarding as for individual with autism as it is in the neurotypical brain. Okay, now switching gears somewhat, but ultimately, we're still staying in the realm of the social brain, of course, and we'll see other findings implicating the reward circuitry, looking at a series of studies that have been published over the past year, looking at the role of oxytocin and the social brain. So here, the first study was quite an interesting study. Again, I don't have time, and I wish I did, to go into the details of it, but they've used similar stimuli to the one that one of my first graduate students used, where they presented the subject in the scanner with information that was, so they had some stories. They saw a person with a face, and basically there was something that was happening. The information was there was either a congruous voice speech with what happened, or there was incongruous. So you can see something bad happening and the tone of voice is positive, or vice versa. And one of the regions that, as I said, my former graduate students and I have found to be really responsive in tasks where you have to make sense of what the speaker really meant, is this ventromedial prefrontal cortex. And what they have found in the study is that indeed, just intranasal administration of oxytocin normalized activity in that region. Okay, and this was a well-done studies. I mean, I promise you, I'm not presenting you anything here that did not pass some minimum standards in terms of method. This was a randomized, double-blind studies within subjects. So there were basically every subject was doing the scan, going through this procedure twice. They were randomized to do it first with oxytocin or with a placebo, et cetera, et cetera. Then there was a second study that again looked at the effect of intranasal oxytocin, but this time focusing on some of the brain circuitry that we know are important for processing information in the faces. And again, they found a significant increase in amygdala activity in response to administration of oxytocin. And my favorite among the series of studies is actually this last one done by some of our colleagues and collaborators at Yale, which actually focused on the entire social brain regions. And here is what some of their findings look like. So this is just to show that their administration of oxytocin actually increased plasma levels, and these are the tasks. They basically used a very well-known task where they were presented with pictures of eyes like that, and the subject had to decide which mental state that picture best depicted. And this is a well-validated study used behaviorally in many other research studies. And then they created a control condition where they were seeing pictures like this depicting a portion of a vehicle, and they had to decide whether it was a sport car or an SUV or a bus, and that was the control. And here are some of the findings. This is again a very well done study, double-blind, placebo and whatnot, and you see somewhat similar responses, but I hope that just by glancing at the picture that you see following the administration of the oxytocin and the one following the placebo, you see that there are differences in the magnitude and extent of the activity Overconnectivity, which when they were directly compared, you saw translated in some significant differences, again, in part of the reward circuitry, as well as in the theory of mind regions, and also some in the superior temporal sulcus. And one of the points that I wanted to point out about this study, particularly with reference to what Jim McCracken's talk today pointed out, is that in this study they also had behavioral data, right? The subject were performing a task. And despite these differences in the level of activity observed when the subject had received the oxytocin, there were no behavioral effects that were detected. Okay, so this is a case where I think it makes a point that maybe we should not be so fast at judging the effectiveness of a drug just because it does not yet yield a change in behavioral performance. I am with you that ultimately what we are seeking to do is to alter behavior to ameliorate deficits, but at the same time, I think this kind of data caution you against throwing away the baby kind of with the bathwater and dismissing the potential therapeutic effects of a drug just based on some lack of a behavioral effect, you know, a lack of an effect. I mean, evidence, what is that expression? I think is a lack of evidence should not be construed as evidence of, what is it? Okay, I shouldn't, absence, thank you, exactly. So, okay, so I take this as comfort and promising data suggesting that indeed more research is needed in this area before we dismiss the use of oxytocin. Okay, so now switching gears to connectivity findings, there was a big splash in the neuroimaging community because some of our colleagues had to basically almost not, they didn't have to retract their findings, but they basically had to publicly admit that some of the data that they have, findings that I've reported, were due to some artifacts. And those data, they per se were not in the autism, and they did not pertain to autism. Those were data that show that younger children, in comparison to adults, tend to have increased local connectivity and decreased long-term connections. Okay, so what this same group found is that residual minimal amount of motion differences between children who tend to move more and adults were actually causing that trend. So you would say, like, well, why is it relevant to us interested in autism? Well, because the same differences that this group had found between children and adults have all mirror what we have been found and many others between children with autism and neurotypical control, meaning study after study in the autism literature have suggested that the brain in the individual with autism is more locally connected with fewer long-distance connection. And so that begs the question as to whether this could be also a problem and an artifact due to this residual motion. And one of our collaborators indeed pointed that out in this this comments in Nature. So that sent kind of like the field in a little bit of a fit, but particularly because there were also other studies that showed, even using a different measure here, looking at diffusion imaging, looking at structural connectivity, again, very small, minute differences in head motion, because we've all known that motion is bad, and we've been trying to control it and enter as a regressor. All of that was above and beyond, on top of what you could do to correct for motion. Anyway, there were other studies that showed that you could see this pattern of artifacts or confounds also in structural imaging. So a lot of us went back and reanalyzed our data doing this using this motion scrubbing technique, which is very conservatively. You go and remove a lot of your data and you throw it away to make sure that it's not impacting your findings. And thankfully now there is more and more papers that are actually coming out showing that even when you can still find some of these same connectivity differences even after you do this fancy motion scrubbing, as it's been termed. So basically, in essence, much ado about nothing. It's not like everything that we have shown is due to artifacts. Nevertheless, it certainly has cautioned the field and made us more aware of some of these methodological issues. So aside from this major concern, or ameliorating also some of these concerns, are some interesting data that have been published again in the past year that are beginning to go beyond merely characterizing differences in connectivity. Basically, the first wave of connectivity studies have basically reported differential activity within different networks in the brain, and now what we see is more and more studies directly relating this changes or altered connectivity patterns to behavioral phenotypes or severity of impairments and so on. And I think I'm going to try and speed up and not give you too many details of each of them for the sake of making it through my presentation in time. But for instance, there was an interesting study here that focused on the networks, the connectivity within language networks, and they found significant relationship between some of the connectivity, both in the language cortices in the left hemisphere as well as in the right, with language communicative deficit. There was another study that looked at the so-called default mode network, which is one of the best characterized networks in the autism literature. It's one of those intrinsic networks that it's implicated in social cognition, which involves both the medial prefrontal cortex and some of the parietal cortex. And here they found something quite interesting, actually. They teased apart this posterior hub of the default mode network, and they realized that some subregions within that area show hyperconnectivity and some region showed hypoconnectivity in the autistic brain, again suggesting that we should be quite careful in making over general statement about the autistic brain being underconnected. And interestingly, again, they also found some significant relationship between the extent to which this hub of the default mode network showed decreased connectivity and social impairment. Okay, and then what I'm going to tell you a little bit more about it. What was my favorite study. Obviously, beauty is in the eyes of the beholder, so somebody else may differ, but this is one of my favorite studies because it focused, it used both task-related fMRI as well as functional connectivity and related both type of measure, both to each other and also to behavioral patterns. And so what they found this region in the superior temporal sulcus, which is hopefully, I don't know if you realize, this is some of the same region that we see in, repeatedly coming up in many studies that look at autism, because it's a region that it's important for the perception of biological motion, which is obviously very relevant to social cognition. So here is the area that shows significant differences in brain activity when they were actually looking at point of light displays. Have you ever seen when they put some lights on the different what do you call it? Yes, joints, thank you. And so then when you can look at this patterns of dots, and I was counting on Scott, who has used this stimuli to show some of those pictures early in the morning, but it didn't, otherwise I would have put it in there. Basically, when you're looking at this dot display, you cannot not infer biological motion. You know, it jumps out at you. And so that's the task. They had to decide what that person was feeling, because different type of motion really can express different emotions. And they found this difference in between the two groups, where the individual with autism showed less activity, but as you can see here, there was a very nice relationship actually in both groups, but stronger in the individual with autism, such that the better the performance on the task and the stronger the activity. Then they looked at functional connectivity. This is the seed region, and they said, where's activity from this region? Where do you have connectivity with this region? And again, you see in purple the areas that show overlap between the two groups, and in red would show greater connectivity in the ASD, and in blue what regions that show connectivity with this seed area just in the typically developing group. And by the way, for those of you who might have been here at a few other presentations, some of these regions are part of the so-called mirror neuron system, or in this case they call it action observation network. Here is again some of the differences, the significant differences between the two groups, again basically mirroring what you've seen there. And then they also related this differences in pattern of connectivity between the regions to behavioral performance, and again found very significant correlation. So I kind of like to, I like the study because it shows how you can, I guess, be confident about your findings when you kind of go full circle and seeing how behavior maps onto both functional alteration in functional activity pattern, how those relate to functional connectivity alteration, and ultimately how those also relate to the behavioral deficit. So I've already alluded to this when I mentioned briefly one of those studies that focus on the default mode network, and they actually teased apart that region and look at sub regions within it. And I mentioned there that there were some regions showing hyperconnectivity, greater connectivity, and some regions showing underconnectivity. This is basically the other, I guess, big story of 2013. Initially, as I mentioned, there were studies after study that showed hypoconnectivity in the autistic brain. There were already a number of studies that went counter. At the beginning, we noticed, for instance, that some of this first study that showed overconnectivity, they found it between subcortical and cortical regions. So the theory was still kind of, people could still try and make the theory of the underconnected brain fit, but as we progress into the year, there were more studies that showed that maybe that at this time we had to rethink this notion of an underconnected brain in autism. So there were two studies that actually came out in the same issue of self-report, and the first one showed overconnectivity in this particular case limited to the posterior occipital cortices in individual with autism, and then a second full article, this was a brief report in the same paper, was actually showing brain hyperconnectivity in a variety of different networks. And the same authors that, of course, they already knew that they were sitting on this data, had just a few months earlier published another review articles where they kind of started tackling with this issue that there were now enough reports finding overconnectivity in the autistic brain that we really had to start trying to integrate and make sense of these findings. And what they have proposed by looking, by comparing the findings across studies, is that looking at taking a developmental perspective could perhaps help making sense, because what they noticed is that most of the studies that found underconnectivity were looking at an adult brain, whereas the studies that found the overconnectivity, okay, let me see that I'm making it straight. Yes, yes, so the notion was underconnectivity in the older adult brain and overconnectivity in the younger brain. Except that, you know, when we were asked, my former graduate student and I, to actually write a brief preview to those two papers, you know, we had read obviously that review paper, and we thought, yes, well, that it's nice. However, we know that there were also studies published by Dinstein in young toddlers with autism that showed again the underconnectivity that was found in the older adults. So you know, when it's hard for me, as much as I'm a developmental psychologist and I like to take development into consideration, I just, when you start seeing a lot of a curvilinear relationship where the brain initially is underconnected, then it becomes overconnected, and then it's again underconnected, I mean, I kind of feel a little bit uneasy, and I begin to suspect that there may be other factors at play. It's basically, in a nutshell, as an oversimplification as it can be. It's complicated, and I think we have not seen the last of studies that will try and tease apart the relative influence of age, which is, I'm sure, an important role, together with other methodological factors. So stay tuned for more. Now, I'll tell you one study that showed overconnectivity. They actually used, the aim of the study was to evaluate whether you could use connectivity pattern in the autistic brain to actually identify individuals with autism from neurotypical control, and also to relate that to symptom severity. And out of 10 intrinsic connectivity networks that they focused on, they found that the group of children with autism was actually showing overconnectivity, greater connectivity in 6 of them. So again, more to the point that some network may show overconnectivity, some network may show underconnectivity. But the thing that interested me the most about this study was that even if they found the significant changes in 6 networks, it was particularly this Salience network that those of you who know me know I feel I'm quite interested. We wrote a grant on it. Was the one where it's actually the only one where you could actually, that could be used to differentiate individual with autism from the others. And this is what they did. They basically use a classification algorithm. They basically classifying each brain, because the beauty about doing imaging is that you can actually get this pretty maps at the single subject level. So you basically get this nice map, and you can feed it into the algorithm and classifying it. And then, you know, there's a lot of more details that I won't bother you with. And ultimately then, after you've trained the system, you present them with new cases. It would be bad if they had done it with the same individuals that were fed into the training part of the study. And they found actually that you could, the system could actually predict, you know, whether a particular pattern of brain, basically whether a brain belonged to an individual with autism or a neurotypical individual, with a fairly high rate of accuracy. Now, this is not something that, you know, in and of itself could be used to make a diagnosis, but this other picture that I have here shows that so the extent to which the Salience network was showed the greatest differences. But what I put it up here to illustrate is another point, and that is that in and of itself, the Salience network was the only one that did a good job at discriminating individuals with autism from neurotypical. But again, it wasn't like he was doing it with 100% accuracy or sensitivity, but maybe, just maybe, if we start combining, since we can get this network from every single individual with just a 8-minute resting state scan, maybe if we start combining the information that is contained in each of, you know, pattern in each network, maybe we can achieve some better numbers. So I think that there is hope for the future, and in particular, I'm quite interested about this line of work because, again, as I mentioned, you can get this network even when a person is asleep, and we are now conducting studies in infants at very high risk for autism, and we scan them when they are in natural sleep. So we are in a position of collecting this type of data, and hopefully we'll be able to use it to inform diagnosis later on. In terms of future direction, very quickly, there have been some, with regard to conventional task-related fMRI, the things that stood out is that there seems to be finally some interest in looking at sensory responsivity, which is something that has not really been studied much in the past. I mean, there was a study just looking at autistic trait and responses to affective touch, and then one of the studies done here at UCLA by one of our fabulous graduate students, Shula Green, has focused on over-responsivity in response to both auditory stimuli that sounded like, okay, so not very, not very fun. And this one, it was the visual stimulation. It's not going to look nearly as annoying, I suspect, right now, but you have to understand that that would basically, that's all they see. This is in front of their eyes, so it's not like a little thing like that. And lo and behold, this is what the findings show. Typically developing kids, children with autism diagnosis, and the significant differences. In this case, the children with autism show greater activity in some of both the sensory regions, but also for those of you who can spot it, in the amygdala, which is obviously a region implicated in emotional processing and negative stimuli, and focusing on this same region of interest in the amygdala, you can see that there is also a nice relationship between the extent to which this region responded to the stimuli and the parental report of over-responsivity to sensory stimulation in both groups. Okay. Very, very quickly, there was another interesting study that looked at, as you can see by the title, it said that the brains of high-functioning autistic individuals do not synchronize with those of others. What was interesting in this study is that they didn't use the typical paradigms where they present you with some stimuli and then another sort of stimuli, and then you compare activity between the two. Every subject watched 45 minutes of basically a movie depicting social interactions. They did not contrast activity during that movie viewing. Rather, they look at correlation between subjects in terms of the pattern of activity. And what they basically are looking is for similarity of responses across individuals to the same kind of social stimuli. And lo and behold, they found that individuals with neurotypical individuals had much stronger pattern of correlation, that is, their pattern of responses to viewing this socially meaningful movies were much more similar than in an individual with autism. And what I found interesting about this is this notion that maybe, just maybe, some similar tools like this could be used to start teasing apart the heterogeneity in our individual with autism. So this study here just focused on differences between neurotypical and individual with autism, but we all know that there is tremendous heterogeneity in individuals who are receiving a diagnosis, and so I wonder whether actually we can start using some of these methods to tackle that issue. Last but not least, I saw this was published this past year what I believe is one of the first studies that looked at biological or gender effects in the brain of autism, and this is something that I promise you will see more of, because Suzanne Buchheimer, myself, and some of our colleagues at Yale, Harvard, and University of Washington are part of an NIH-funded autism network of excellence that is designed to actually look finally at differences in the male in boys and girls in our study. It's actually a developmental study, so we're looking at boys versus girls. And last but not least, I'm going to finish in time this time. I wanted to also highlight that there is strength in number, and what I mean with that is I think this is a remarkable achievement of the past year, although we actually started putting our data together before this paper came out. This is the ABIDE imaging data exchange that at this point includes more than 1,000 brain, where institutions, investigators from all over the country and abroad, they have volunteered their data, and everybody can access this data and its resting state, functional MRI, together with a lot of phenotypic measures that can be used to now try and make sense of some of the discrepant findings that are in the literature, because let's face it, methodological differences play a role. Sample characteristics obviously play a role, but also sometimes you have to keep in mind that when you use a very limited sample size, some of it may also be noise. And so on that note, I will thank you for your attention, and I thank you also, my collaborator, the funding agency, and the children and parents who participated in our studies. Thank you. We were talking about the last study that I was mentioning about the over-responsivity. In that case, they were all, I mean, the kids with autism were showing there was a correlation with the parental reports, but overall, the group of children with autism that showed greater responsivity to sensory information by the parents' report also showed over-responsivity in the brain data. So there we did not see hypo-responsivity. So I'm not saying that you couldn't see it in another study, but in that study, given the nature of those stimuli, they had more of some greater responses in terms of neurobiological responses that we could map.
I was just wondering if you found any abnormalities on DTI or functional MRI with pathways from the cerebellum to the frontal cortex?
In our data, I believe that some, in the paper, the graph theory, a recent paper that we published in Neuroimaging Clinical, there were some pattern of alterconnectivity that involve also the cerebellum, and I suspect that that was not the only study, but it's not something that it's jumping out from the literature. And I think in part is because you know, we now know that the cerebellum is playing a much more active, you know, important role in cognition and in a variety of different tasks, so that it may not necessarily show up as uniquely contributing to one of these intrinsic network or another. If it's involved, you know, in many different functions across the board, and in a way, it may be actually harder to disentangle its specific role.
What's the function of that salient network?
Yeah, the function of the salience network, by the name that it was given to it, it's supposed to be basically a network that switch or, you know, in a way allocate attention to what is most salient for the individual, and in some cases it would be something that is happening in the environment. So if you're a baby, you know, say your mom and dad and whatever, it's happening around you. But at the same time, also when you're a baby, when you're hungry, you're going to cry because there is something internally that, you know, you're not feeling good, or if your diaper is wet. And so there are people who believe that the role of this network is to basically switch to kind of either focus on what's out there or focus on internal states. And so it's supposed to modulate activity of one of the dorsal attentional network, which is typically, or the executive cognitive control network, which is focused on what's happening out there, what demands my attention in the environment, and the so-called default mode network, which is important for social cognition, but more from an introspective point of view. So in a nutshell, it's supposed to really direct the attention as to what is most critical in your environment, whether it's here or that. So I suspect that it's going to be we focus on that because we think it makes logical sense that this be a network, that it's fully functional very early in development. Thank you.